Okay, so we have here the catalase negative na gram-positive bacilli. Okay, the first one, we have your erysipelotrix rochopathy. Erysipelotrix rochopathy can be found in the environment. Most likely they are found here in vertebrates or invertebrates, including here the swine, the birds, and even the fishes. So most likely human infection is by close handling with contaminated na mga animals. Most likely this one is occupational, like those who are having the work related to handling the fish or even your poultry products. Okay, so erysipelotrix infection, the human try to cause here three main manifestation. The first one, we have the localized skin infection. You call this one your erysipeloid. That one could resemble here the erysipelas of your strep. Then the second one, we have your sepsis. That may be in the form of your bacterial infection, sepsis in the form of your even your endocarditis or inflammation of the heart tissues. And lastly, we have here the diffuse generalized na skin infection, cutaneous infection. So more of the skin infection ito. Again, they could enter your body by a cut on your skin, abrasion, or even the breaks in your skin. Okay, laboratory diagnosis. So we have here the microscopic identification. The gram stain result of your erysipelotrix appears to be a thin filamentous gram-positive bacilli that may appear in single or short chains or even assume here the V shape, just like our Corynebacterium diphtheriae. This one is easily washed off by our decolorizer that might have here a gram variable staining reaction. Okay, we have culture here, so able to grow in the SBA within 24 hours that appears to be beta hemolytic. But that would be a pinpoint na colony sa sobrang liit niya. But after 48 hours, pag maging age na tagal na ang culture natin, this one would try to produce here dalawang culture characteristic. Number one, the small na colony appearing to be smooth, glistening, entire, convex. The other one is large. Meron siyang large na colony, but that one appears to be rough, irregular, with curled margin. And that appears eventually to become here alpha hemolytic. So, pag 24 hours is beta hemolytic, pero after 48 hours, pag tumatagal, nagiging alpha hemolytic siya. Mean to say, it become here alpha hemolytic is partial hemolysis na lang siya. Okay, identification here. So, this one is catalase negative. This one is non-motile. It will have here very prominent na characteristics with H2S positive. This is the only one here in gram-positive bacilli natin na H2S positive or hydrogen sulfide positive. Then we'll have also here, have a characteristic na test tube brush na colony growth here in our gelatin stock at 22 degrees Celsius na growth temperature. Okay, the next one, na catalase negative, we have here your Arcanobacterium hemolyticum. Arcanobacterium hemolyticum try to cause here 4 infection. The first one is pharyngitis. So pharyngitis, they resemble your strep pyogenes infection. So parang sore throat, diba? Sore throat is your pharyngitis. We have also here your cervical lymph adenopathy. So inflammation, swelling of your cervical lymph nodes. We have also here the discrimination of your hands and feet. And we have also here your soft tissues, including your sepsis, endocarditis. Okay, then we have here the lab results. So the gram stain results of your Arcanobacterium hemolyticum appear to be pleomorphic. So again, it would assume here different shapes. But ang pinaka-striking na morphology nito is rudimentary branching form. And it also appears here to be cocci, more of the cocci after 48 hours incubation. Cultures are able to grow here in the SBA. This is beta hemolytic with a narrow zone with a pinpoint na colony, so sobrang liit din. And this one is capable of producing also the black opaque dot. If you try to scrape the agar, so it would produce a black opaque dot, para siyang may black dot. And also try to pit the agar. Pag sinabing pit the agar, so parang magkakaroon siya ng depression. Okay, so this one is capable of pitting the agar. Okay, then we have also here the identification. So this one is lecitinase and lipase positive. Lecitinase ang positive natin that will be manifested with your egg yolk agar, and lissadinase positive is characterized by opaque zone or the clear zone around the colony, opaque zone. And we have your lipase, parang siyang sheen, parang siyang gasoline, parang gano'n makikita natin. Another one, this also contains the phospholipase D. Okay, the phospholipase D is the substance produced by this one which try to inhibit the beta-lysine produced by your Staph aureus. Therefore, pag na-perform tayo ng CAMP test, di ba, usually we are streaking for the Staph aureus. So, pag may Staph aureus ang ginamit mo, tapos streak mo ito perpendicular to your Staph aureus, wala kang ma-form na hemolytic reaction. Because again, the beta-lysine produced by Staph aureus is inhibited by the phospholipase D of your Echinobacterium. Pero pag ang streak natin is your Strep agalactiae, tapos streak mo perpendicular to that, your Echinobacterium able to produce here hemolytic reaction. And therefore, you call it once your reverse CAM test. So instead of the Staph kasi ginamit natin, so Strep agalactiae ang ginamit natin, making this one as your reverse CAM test. Okay, then the drug of choice for that, okay, so would be your erythromycin. Okay, the next one, we have your Gardnerella vaginalis. So, the main clinical infection associated with this bacteria would be your bacterial vaginosis. But your bacterial vaginosis not only caused by your Gardnerella vaginalis but also being caused also here by other bacteria like your Mobiloncus, Prevotella, Peptostreptococcus, or even your Mycoplasma hominis. So, bacterial vaginosis is characterized by the pH, vaginal pH more than 4.5. Usually kasi normal, dapat slightly acidic lang siya, 4.5 ang vaginal pH. Pag tumaas ang pH niya, it would result here to your bacterial infection where it would have your primarily populated by your mga ganitong bacteria, even your Gardnerella vaginalis. Your bacterial vaginosis also characterized by the decrease in your lactobacillus. So, lactobacillus maintain the normal pH in the vagina. Okay, this is characterized by malodorous discharge. So, sobrang baho ng discharge niya. Okay, lab result here. So, gram stain result. This one is pleomorphic, appearing to be Coccobacilli. And that one would have your, okay, eventually identified also here by your Wetman preparation with your vaginal secretions for identification of the presence of your clue cell. Clue cell is just a squamous epithelial cells wherein the entire surface ng ating squamous epithelial cells being covered by your bacteria. So, kung ito ang squamous natin, okay, lahat dito, throughout that, may bacteria. That's your clue cells that would identify your Gardnerella vaginalis infection. Okay, culture we have here in the SBA, so it appears to be pinpoint non-hemolytic. But it's ideal na culture media for selective na culture media for identification for the growth of your Gardnerella vaginalis would be your human blood bilayer twin agar. So on that, it appears to be hemolytic siya, beta hemolytic, appearing to be gray, small gray and opaque na colony. Pero pag nasa SBA lang tayo, that would appear to be non-hemolytic. Okay, this is also blood agar din kasi ito eh. Okay, then we have here another presumptive test for identification of your Gardnerella vaginalis infection. You can perform here the whiff test. With your vaginal secretion, you add 1 drops of your 10% KOH, tapos amuyin mo. Okay, positive result will have your fish-like odor, so malangsa. There's a positive result for that. The best way to diagnose your bacterial vaginosis by Nogen scoring. So, on the Nogen scoring, you try to quantitate kung ilan ang concentration ni Mobiloncus, ilan ang ating Lactobacillus, and ilan ang ating Gardnerella. So, para siyang quantitative estimation. So, give the score, gano'n. Normal scoring is 0 to 3. So, that's normal vagina, normal. Pero pag ang score niya is 7 to 10, okay, that will identify na meron siyang bacterial vaginosis. Okay, Okay, the main differentiation with your gram-positive Bacilli here would be the catalase test. So catalase test is would identify the enzyme catalase that would convert here the substrate hydrogen peroxide to your water and oxygen. The positive result will be the bubble formation. So this one will give a positive result only for your Corynebacterium and your Listeria. Again, that will give a negative result for your Erythrophilodrix, Arcanobacterium, Gardnerella, and even your Lactobacillus. Okay, we have here the summary of the entire characteristics of your gram-positive non-spore former, non-branching na mga bacilli. So, we have your catalase test. Again, it will only give a positive result with your Corynebacterium and your Listeria. Motility at 25 would only be with your Listeria because this one is capable of your end-over-end tumbling motility. Bile-esqualene hydrolysis will only be with your Listeria monocytogenes that would resemble your group D na strep. H2S in your TSI, triple sugar ion agar, is only manifested in your Erythrophilodrix rostropathy. Again, H2S hydrogen sulfide would be characterized by the blackening of the colony. And the urease test is only positive here with our Gardnerella vaginalis. Okay, now we go to your branching form of your gram-positive bacilli. We try to form here try to branch. So this includes your actinomyces and even your nocardia. First one, we have here the nocardia. Okay, nocardia can be found here variety of environmental origin. It could be found here in the soil or even in your plant materials. Okay, so the following are the virulence factors: superoxide dismutase, catalase. Another one is your nocobactin. Nocobactin is the iron chelating agent. We try to chelate the iron of the host and eventually try to deprive the host here with its own iron supply. Another one, clinical manifestation or the infection caused here by nocardia. So, this is very common among immunocompromised na patient. There are two forms of the infection caused here by our nocardia. One is your pulmonary in the form of your pneumonia. Okay, and that one differ with the other na mga pneumonia because number one, this is just a mild inflammatory process. Within mild inflammatory scarring, so mild lang siya. Another one, wala siyang granuloma formation, wala siyang sinus tract formation, wala siyang sulfur granules, and wala din siyang encapsulation of your abscess. The cutaneous, on the other hand, is caused here by Nocardia brasiliensis, so it's a skin infection, and you get infected here by, again, the cut or abrasion in your skin that would allows the Nocardia in the soil to enter your skin. So, pag skin infection, it would be in the form of your lesion, which try to affect the subcutaneous tissues. You call that one as your actinomycotic mycetoma, which later on, pag hindi siya nagapan, the lesion here try to worsen, which try to break open. So, lalabas na may nana na, and the nana here is yellow-orange. That's why tinawag siyang sulfur granules. Okay, then we have here the laboratory diagnosis for the nocardia. So, first we have here the microscopy, the gram stain result. Okay, so this one appears to be beaded, long branching bacilli. Then eventually that would have here considered as partially acid fast. Partially acid fast because it's made up only of the short chain of your mycolic acid. Hindi talaga siya acid fast bacilli compared with your Mycobacterium tuberculosis. Because again, the mycolic acid here is just a short chain. And that one, you are using a weak na decolorizer, that's 0.5 to 1% na sulfuric acid. Pag ginamit mo dito is the acid alcohol with your acid fast bacilli staining reaction, magiging gram-positive ito or magiging color blue ang color nito. But you are needing here your weak na acid lang, 0.5 to 1% na sulfuric acid, that would render that one to be decolorized. Then pag-add natin ng ating counter stain, which is your methylene blue, hindi na siya ma-decolorize. Okay, and therefore, that one would retain the primary stain na color. It will give a red na color. Pag acid fast kasi, ma-decolorize siya. So, pag pag acid fast with the use of your acid alcohol, ma-decolorize siya ng acid alcohol. Pag nag-add tayo ng methylene blue, magiging blue ang color nito. Therefore, acid fast lang siya, partially acid fast with the weak acid na 0.5 to 1% na sulfuric acid. Okay, culture here, so it will able to grow in your SBA, appearing to be na beta hemolytic. The best culture media for that, you can isolate that one using your modified thyroid Martin for your Neisseria na culture media. Your buffered charcoal yeast extract could also grow on that. Mag-grow din siya sa ating mga fungi na culture media like your sabarode agar or even your mycocell. Ano naman magiging tsura niya sa culture media natin? So parang fungal-like ang kanyang characteristic. It appears to be like velvety, okay, matte, chalky, dry, powdery. So parang siyang breadcrumbs. So parang siyang fungi natin. Okay, that one we're able to grow at 22 and 37 degrees Celsius incubation. That's for 3 to 6 days. Medyo matagal siya mag-grow. Then we have also here the other genus of your actinomyces. So we have your actinomadura. This one would have your moderate na intertwining branching na form. Another one, we have here your streptomyces. That one would have an extensive na branching. This one is moderate lang sa kanya. Sobrang branch talaga. Gordonia appears to be short-rod. Schucamorrhea would have your long na bacilli with striata fragments na puputol-putol. And we have your rhodococcus appear to be cocobacilli zigzag na form. So of all your other actinomyces, Gordonia, Schucamorrhea, and rhodococcus are also partially acid fast just like our Nocardia. And then we have also here your Tropherema weeflate, which is the causative agents of your Weebles disease. Okay, so that's with our spore form, that's all with our branching form na gram-positive bacilli. Okay, now we go to the spore forming gram-positive bacilli. So they have the spores. Again, we have two types of spore, the endospore and the vegetative spore. Okay, the endospore, the one which is the dormant, inactive, but it could be converted to become vegetative once they are exposed to your harsh environment. So we have here the genus Bacillus. The first one, we have here Bacillus anthracis. So this is the bacteria isolated by Robert Koch among the cow. This one try to cause here your anthrax. Okay, so this contain capsule as a protective na barrier against your phagocytosis. The capsule here would have a very special na component in the form of your polysaccharide D-glutamic acid. This also produces here toxins, which are usually they try to combine with one another in order for them to become toxic. But individually, they are not toxic. The first one, we have your protective factor. So the protective factor try to bind with your edema factor and even your Liddle factor in order for that to become toxic. Edema factor, once combined with your protective factor, that try to cause your edema or accumulation of the water because of the increased na cyclic adenylate cyclase, increasing your cyclic AMP, which allows the accumulation of your water resulting to your edema. Then we have your Liddle factor, once combined with your protective factor, try to cause your death of your tissues. It try to inhibit here the transduction of your signal regulatory protein, and that would result here to the death of your tissues. Again, each of this one become here toxic once they are combined with your protective factor. Okay, then we have here the different clinical manifestation of your anthrax. The first one, we have your cutaneous anthrax. Cutaneous anthrax is just a skin lesion because of the entry of your spores in the skin through breaks or open wound in the skin of the patient. So this is characterized started with kung saan siya pumasok, nagkaroon siya ng lesion, para siyang pimple-like, pustule, which eventually nag-spread magiging erythematous ring. And later on, it try to dry, ulcerate, magdadry siya, ulcerate and dry, and try to produce here your black S-scar. Black S-scar, ito yung parang scar niya na may black center. Okay, you call that one as your malignant pustule. Actually, hindi siya malignant, hindi siya pustule. Because this one is painless, and at the same time that one, hindi siya nagkakaroon ng pus forming, or wala siyang pus, or WBC. Wala siyang nana. Another one, inhalation anthrax. Ito yung, di ba, sa powder form dati, nagiging agent of biological weapon ang ating anthrax because of this one. They try to send mga meals. Pag open mo, may powder, then you get infected by that. So this is also called here wool sorter's disease. This is very common for those na mga handling animals. So, mga fur ng animals. So, ganun siya. Kaya inhale. You try to inhale that one. Okay, so this is a respiratory infection characterized here starting with a non-specific illness like fever, headache, sore throat, malaise. So the first few days become here para siyang mild phase. But then again, it can progress to become severe phase. The severe phase here meron siyang respiratory distress. So that means na hindi makahinga ang patient natin so pwede siyang mamatay because of that. Gastrointestinal anthrax on the other hand is by ingestion of the spores. This is the most severe. Bakit siya most severe? Kasi most likely ang manifestation na is gastroenteritis lang. And nami-misdiagnose natin siya. Because by nature, di ba, sinabing anthrax, it's more of respiratory. Hindi siya common na causative agent ng gastroenteritis. Kaya so usually nami-misdiagnose siya and eventually hindi siya natitreat ng maayos. And that's where nagkakaroon ng complication and therefore considered as most severe because of that. Okay, another form of your anthrax infection, we have here your injectional anthrax. Injectional anthrax is just your injection of the drugs directly to the skin of the patient. Or you call this one your popping, skin popping. Or this your causing your soft tissue injection that would resemble here just like our cutaneous form of the anthrax. Only that the pinagkaiba nito ay wala siyang black scar formation and this one's more severe compared with your cutaneous anthrax infection. So we have here the lab diagnosis. So gram stain result, it will appear to be this is your square-ended rods or square-ended na bacilli, square-ended that appears to be in chain that would have your characteristic na bamboo pole appearance or bamboo rod appearance or bamboo pole appearance. The spores usually could be found here in central area or even subterminal. Okay, we have your colony characteristics. So in the SBA, this one appears to be non-hemolytic. It's large, flat, and gray, and it would have here irregular projections or filamentous projections. Kaya para siyang, di ba, projection parang ganun. May mga extension that gives it a medusa head na characteristic na colony. This highly mucoid, wherein pwede mo siya pag-inflict mo ng loop ang ating culture niya or colony niya at able to stand straight. Kaya na parang stand straight, just like parang siyang beaten egg white characteristics. Then we have here the string or pearls test with your penicillin susceptibility testing with a 0.05 to 0.5 unit per ml of your penicillin antibiotic. On this one, your Bacillus anthracis appears to be susceptible. Meaning to say this, your Bacillus anthracis is inhibited by your 0.05 to 0.5 unit per liter of your penicillin. And that we consider that one as effective against your Bacillus anthracis. Okay, the next species of your Bacillus, we have your Bacillus cereus. Bacillus cereus here try to cause your food poisoning. So there are two forms of food poisoning. So one is your diarrheal, the other one is your emetic. So we have your difference of the two. So diarrhea is most likely because of ingestion of your meat products or even your pasta or even your dairy products. Emetic would be with the ingestion of your fried rice or boiled rice. Duration, so main symptoms of your diarrheal would have your diarrhea and emetic would have your the vomiting. Then we have also here the heat stability. So for emetic, that one is heat stable. Kaya nga even if you try to heat your rice or fried rice, so still andun pa din siya. Hindi siya mamamatay and therefore try to cause your food poisoning, making this one as your stable, heat stable. Whereas your diarrhea on the other hand is being killed by our heating process. Okay, so this one would have your heat labile natin siya. Okay, then we have here the laboratory diagnosis. So microscopy, we have here the gram stain result. Okay, so this would appear, Bacillus cereus would also same characteristics with your Bacillus anthracis. That one is gram positive square-ended rods or bacilli with the spores on that present either central or subterminal. Colony, it would just differ on this one with the Bacillus anthracis because this one is beta hemolytic in the SBA, whereas your Bacillus anthracis is non-hemolytic, di ba? So, this one appears also to be large, feathery, spreading, would have here the frosted glass appearance. Okay, then we have here the summary of their characteristics. All the species of your Bacillus here are all lecithinase positive. Ang moti lang natin includes your Bacillus cereus and thuringiensis. Your penicillin sensitivity, this is your sting of pearls test. So, ang susceptible lang natin would be your Bacillus anthracis. All the rest are resistant. Paraspora Crystal could only be found in your Bacillus thuringiensis. That's why ginagamit ang ating Bacillus thuringiensis as an agent of your biological insecticide. Because meron siyang parasporal body. Then we have here some biochemical reactions for the diagnosis of our bacillus. So first one, we have the catalase test. So the catalase test will identify only here the Corynebacterium diphtheriae and your listeria, characterized by the bubble formation. All the rest ng mga species natin, with your gram-positive bacilli, they will give a negative result with your catalase. The lecithinase test in the egg yolk agar will give a positive result with the opaque zone surrounding the colony, and that will give a positive result for all your bacillus. Then motility test in a semi-solid na media, semi-solid na culture media. So this will be identified here by the spreading of the colony from the area where you try to inoculate. Kung dito ka na area na inoculate, like for example pa ganyan, usually tube. Kung dito ka nag-stop, so parang lumipat sa ibang area, that you're positive for the motility. So, moti lang natin, we have the cereus and thuringiensis based on your table. Then, penicillin susceptibility will identify your Bacillus anthracis, that one is susceptible, affected by penicillin, is effective for the penicillin. Effective ang penicillin natin for Bacillus anthracis. All the rest, they are resistant for your penicillin. Another one, we have your parasporal body or Paraspora Crystal could only be found here in Bacillus thuringiensis. This is a special structure considered here to be a protoxin that once ingested by your insects, especially your lepidopteran na insects here, would able to, once inside the body of the insect, it will become toxic and it will kill the insects. That's why since ang Bacillus thuringiensis lang meron nito, ginagamit natin ang Bacillus thuringiensis here as our agent of biological insecticide. Ginagamit siya as insecticide agent. And lastly, we have here the differentiation between your Bacillus anthracis and your cereus. So, beta hemolytic ang ating cereus but not your anthracis. Sensitive ang ating Bacillus anthracis with your penicillin, mean to say that one is effective but not with the cereus. Gelatin hydrolysis and growth in your phenyl ethyl alcohol is only for the cereus. It will not allow the growth of your Bacillus anthracis. Okay, so that's all about our gram-positive bacilli. Thank you.